Hello, this is Nick Oswald welcoming you to this bite-sized bio web seminar, which today is sponsored by Novus Biologicals, who provide antibodies, proteins, peptides, RNAi, lysates, stem cell lines and antibody labeling kits to the biosciences community. You can visit them at www.novusbio.com. Today's presentation is titled Chromatin Immunoprecipitation, Struggles and Solutions, and is being presented by Dr. Eric Neely, who is Lab Director at Novus Biologicals. As always, we will have a question and answer session after the presentation, so please type any questions that you have into the questions box, which appears on the right-hand side of your screen, and I'll put them to Eric at the end. And so now, over to you, Eric, for the presentation.
Thank you very much, Nick, and thanks everyone for joining us today. As Nick said, today I'm going to be talking about chromatin immunoprecipitation, or CHIP, and I'm not going to go through all the specific details of the CHIP protocol, but instead I'm going to focus on the problems that occur either when starting CHIP or ways to optimize your CHIP assay if you've been doing CHIP for a while. So just basically, what is CHIP? CHIP is using antibodies to look at DNA and protein interactions. Normally, when we're doing any sort of immunoassay, our end reporter is looking at the antibody activity and looking directly at the protein. Whereas here, that is just an intermediary step, and our end step is actually looking at the quantification of DNA to determine specific locus or loci that the protein or proteins were binding to. So in the end, we can analyze and measure this by PCR, quantitative real-time PCR, doing a chip-on-chip, which is doing a chip followed up by a microarray, or doing a chip-seq, where we're doing a chip followed up by actually sequencing the product. So there are 5 rough phases of CHIP that we're going to go over here today. The first one here is cell fixation and cross-linking your proteins to your DNA. That is followed up by a shearing or fragmenting of the DNA, then actually immunoprecipitating the protein with the DNA cross-linked to it, then reversing these cross-links, purifying the DNA, and then finally measuring and analyzing the DNA in the end. So there are 2 different types of ways that you can first start off in phase 1 here in looking at the CHIP, something called a native CHIP, where you're actually not cross-linking the DNA at all. You are looking at the native binding of the protein to the DNA, and this is usually used for very strong interactions such as histone antibodies that have a very strong binding kinetics to the protein, versus an X-CHIP, where you actually are cross-linking the DNA to the protein. The other main points that I'm going to talk about today in these other phases are the DNA shearing phase, doing either sonication versus enzymatic shearing. In the immunoprecipitation phase, talking about different bead technologies that exist for pulling down your proteins. Talking about purification methods and different ways of doing this, and finally real-time PCR methods. And we're going to focus this talk today on real-time PCR as being the end method of quantification. So first of all, the DNA shearing phase here, phase 2. One of the most common troubleshooting phases of CHIP is this phase here, in properly optimizing your fragmenting of your DNA. So what we have here, if we look at this first picture, is a ladder going anywhere from 100 base pairs to 3,500. And what we want is our DNA to be sheared roughly from 100 to 1,000 base pairs right here. And you can see this second lane right here has optimal shearing of the DNA that is right in that range, versus the lane 1 here, most of our DNA is clustered around fragment sizes of about 3,500, and then down here we can see that we have grossly oversheared our DNA, and we have too much between the 100 and 200 base pair range right here. Now this is with sonication. So when we use sonication, we're going to see a nice even smear for the most part up and down our gel, versus enzymatic shearing that, although it's a little bit more gentle to the cells and the chromatin, with enzymatic shearing, you're going to see more fragments that are without the smear, because they're going to be cutting in the exact same place every single time. The sonication is a little more random in its shearing, and so you're going to get the smear. If we look at a 3D spectral graph of the exact same gel here, it really, really shows the differences between the shearing states here, where we have our under-sheared DNA grouped up here at 3,500, our over-sheared peaking down here at 1 to 200, and then here we see a nice even fragment smear between 100 and 1,000 base pairs here in our second plane. So what makes the difference when we're sonicating to have these different shearing patterns? Well, number one is the equipment. Whatever you decide to use as your sonicator, whether it be a microtip sonicator, a horn cup system, a water bath sonicator, they're all going to give very different results that you're going to have to play with to see which is going to be the optimal method for you. So the type of equipment matters, the power setting of the equipment matters, how long you are sonicating for, and you never want to sonicate for too long, especially with the microtip type system, because you're going to generate a lot of heat from the sonication. And this heat is going to start to denature your proteins and therefore ruin your binding of your antibody later on. Also, your method, as I discussed, so using a series of short sonications is much better than doing one very, very long. Sonication is going to generate the heat. The type of cell or tissue that you're using for this is also going to be very assay dependent. That will need to be optimized. Your cell density is going to affect things, as well as your fixation time and the efficiency of your fixation. So if you are over fixing and leaving your formaldehyde or whatever fixative you're using on for too long, it's going to create much more difficulties in getting an even shear. And if you are under fixing, you may have the opposite problem. So now we move on to the actual immunoprecipitation step, which is step 3. And by far, one of the most important reagents in the chip assay, of course, is the antibody itself. So most chips are not, most antibodies are not sensitive or specific enough for the chip assay. In sensitivity, we mean the ability to actually bind the protein of interest in a great enough quantity for quantifying at the end, and this is very, very important, especially if you are going on and doing something like a chip-seq procedure to be able to pull down enough DNA for sequencing it later on. And then also for specificity. You don't want to do all these experiments and find out that the antibody that you are using is actually pulling up something else too, and so you either have false positive results or results that are blurred with picking up more than you should. So here we just see this is a selection of antibodies we have for the H3 and H4 histones here. And so let's say we're looking at K4-ME3 modification here. We wouldn't want it to also pull up ME2 or ME1, or at the same time be pulling up ME3 from lysine 9, which is right next to it. So having an antibody that is chip validated is very important. If it already isn't classified as being chip validated and doing the testing on your own, the first step is usually doing a normal IP with a Western blot. If you get fairly good results with pulling down the antibody in a normal IP, there's a good chance that's going to translate well into the actual chip experiment. So now to talk about beads, also a part of the immunoprecipitation procedure here. Here we're looking at just a picture I pulled. It's a slurry of magnetic beads here. And here we're looking at the beads under the microscope at 40x, and you can see they're very uniform spheres of roughly the same size. Now, most people that have been doing chip for a while are used to using agarose beads, and so I'm going to talk a little bit how magnetic is better and may help your results. So number one is saving the time without having to do the centrifugation steps. With agarose, after each wash, you have to pellet the agarose in a centrifuge, then put your pipette tip in and try to pull out all the liquid that's in there without either pulling up the beads and disturbing the beads, or the opposite, leaving too much of your buffer in the tube. So with magnetic beads, since you're separating the beads by pulling them to the side with a magnet, and therefore able to get your pipette tip all the way to the bottom, pull all the liquid out, and not pull any of the beads out. There's also no blocking step. With agarose, typically you have to block with something like salmon sperm DNA or the like, so that you cut down on your background. With the magnetic beads, there's no blocking steps, so you're saving time there. You're also using less antibody overall, since the magnetic beads have a much more uniform coated surface. You don't have to over-apply antibody like you do with the agarose beads that have many pits throughout them that you have to fully coat with antibody. Otherwise, you're going to pick up a lot of nonspecific background too. So you can use much less antibody and therefore save money with the magnetic beads versus the agarose. The better separation and recovery, as I was talking about, it's much easier to separate with the magnetic stand, and it can be automated, yielding better results. So now I'm going to quickly talk about this graph right here. I'm going to be using this example throughout most of the webinar here today. And so I want to make sure that everybody, if they've never really looked at a chip data graph before, knows what they're looking at. So with this example here, we are taking our positive control antibody, which in this case is histone H3 K4ME3 modification, and running it through the chip. So of all the controls that we need for this graph here, the first thing, of course, we need is an antibody. The next thing we need is input fraction. So the input fraction is how we're going to quantify in the very end what our signal of our IP to antibody is to the total amount of DNA that we were to pull from our cell or tissue source in the very beginning. So you'll see here that our number here on the left is signal relative to input. And so for our two bars here, we have about 11% and 29% bound to it. So what that means is during the immunoprecipitation phase here, when we're looking at our input, we're taking all the DNA that was in it. So whether it's bound to a protein that was bound to an antibody or not, like we see here and here, it will even include this section here where there was no binding of the antibody to the protein. And we're comparing that in a ratio to the ones that actually were bound. Our next control is to have a no antibody control, or an IgG fraction only control. And this is just a look at the relative background of your chip to make sure that you're not getting a lot of unspecific binding to your beads overall. And we can see those barely in these little purple bars down here at the bottom, which constitute the no antibody category. So we can see this is good, that when we didn't put in any antibody, we didn't have any binding, so we didn't pull down any DNA to amplify in the end. It's also good if you know the loci of your positive control antibody to have a positive control locus. So here we know that H3K4ME3 is associated with active transcription in cells. So we have two different primer sets here that were designed around constitutively actively transcribed genes, GAPDH and RPL30. And so we see nice amplification of those. In addition to that, we also have four negative loci here that correspond to inactive gene groups and also to heterochromatin, which are not associated with this modification here. So it's nice to see that they did not amplify. So looking more at the beads here, I'm going to have a lot of these comparisons in the webinar here today, where we actually took different components from different manufacturers and tested them to see how they compare. So with the first test here, we're looking at the same antibody, the K4ME3, with that RPL30 primer set here that we were looking at before that amplified so well. And we're looking at it over 4 different bead types. We're looking at it over different conditions, either washing or not washing the beads before using them, and just looking at the quality of the beads overall. So quantity, conditions, and quality. So first of all, if we look at our bead number 1 here, where we didn't wash the beads beforehand, we don't see as good of amplification. And the reason I'm including that condition in here is over testing many companies' beads, I noticed that some had the step in there in their instructions to wash the storage buffer off of the beads versus others not having that bit of information in there. And I noticed with all of them that washing the beads beforehand in whatever sort of dilution buffer you're using for your cells greatly increased the amplification and the binding specificity after doing so. Increasing the quantity of the beads, so going from an incubation with 25 microliters of beads to 50 microliters of beads, also increased amplification, but not as much. And we could see about a 5% increase here from doubling the amount of beads. And this will also occur when increasing the amount of antibody. If it is a specific and sensitive antibody, increasing the suggested amount that's on the data sheet may yield you a little bit more, but it's not going to double or triple your results. And then here with beads 2, 3, and 4, we can see just some very low quality beads that did not amplify this primer set well at all. And these are beads that are very similar. They're all paramagnetic beads. They're all uniform in size, about 2 to 4 microns each. So this is a very apples to apples comparison, but the end results are very different. Now, if we, instead of looking at this positive loci here, and we look at this some negative loci, we see that first of all, the scale has changed. So instead of going to 40%, we're now just looking at 0 to 1%. So these, again, for this modification, should not amplify well. When we look at bead 1 with no wash and 25 microliters, we see very little amplification. Washing them did increase a little bit the amplification of this negative locus, but we're still under 0.2%. And then doubling the amount of beads in there also increased it a little bit, but we're still about 0.3%. When looking at the other 3 beads that didn't really pull down anything before, we can see that some of them are higher. And if we look at one of the other locus here, the alpha satellite heterochromatin, that again should not amplify, we see the exact same pattern as we did with our AFM here. So first of all, looking at your beads and whatever you choose to use, of course, you want to use positive control antibody that you know works well and works well for a specific primer set. But it's also very, very important to include in this the no antibody control to make sure that it's amplifying what it should well, but also not amplifying and not picking up what it shouldn't. So here, if we look at a graph of a real-time PCR run where we see over cycles here versus baseline fluorescence here, we can see 2 different beads that were pulling down the exact same antibody compared to each other, number 1 and number 2. And we can see that they're very, very close to each other. These are both crossing the baseline threshold here at about 22.73 cycles and 23.07. So very close. They're amplifying almost exactly the same, pulled down the same amount of that modified protein, but it's not the full story. When we actually include our no antibody control into this, we still have our traces of bead number 1 and bead number 2, but now we also have our no antibody traces where it was just the beads alone incubated with the chromatin, but no antibody in between to bridge them. So 1 and 3 are matched up, and 2 and 4 are matched up. So if we compare bead 1 with antibody without any antibody, we can see that our cycle difference here is only about 1.4 cycles, which is a fold change of 2.6, which may seem like a lot if you are not comparing them to good beads. If we look at bead number 2, which is a good bead, we can see that now we see a difference between cycles between 2 and 4 of almost 10. And this is now a fold change of almost 900, and percentage-wise that equals about 87,000%. So at first, whereas these beads didn't seem too different from each other, they're both pulling down what we think might be the same thing. They're really not. This one, bead number 1, is pulling back much, much more unspecific protein that it should not be binding, and so you want to see this large, large separation between your bead with an antibody and incubated without an antibody. So another proponent of the magnetic beads here is the option to automate it. Normally we would use a stand like we see here to put the tubes in, and then the magnets pull them to the side so you can separate them. But you can also use a machine that does this for you and use a hands-off approach. The advantage of this are, of course, it's faster to do. It's very consistent and reproducible. You can run the exact same antibody over and over and over again and get the same results, very consistent. You can multiplex. This machine here can do 16 samples at once. So what's happening is these magnetic rods come down and can be turned on, bind your magnetic beads, move them over to the next well, and then wash them. You can also control the stringency of the washing with the machines, much more so than with a shaker or rocker. You can have these come down either very slow and use a speed like this, or they can go very, very, very fast and have very stringent washing. You're also reducing contamination. Since you are not there for every single step, and for this chip procedure here, we've got at least 6 different wells with 6 different steps with your different concentrated salt buffers and your elution buffers, that every time you're obviously interacting with those tubes, you could be contaminating them. And these machines can also be used for purification assays if you use that in your lab. So now we're on to the next step. We've cross-linked our DNA, we've sheared our DNA, we've done the actual IP. Now if we did do the X chip, we have to reverse that cross-linking so that we can take the protein antibody off the DNA and then purify the DNA. So if you look at different protocols with different manufacturers and online, you'll see a lot of differences between how to do this. And usually the variables are the amount of time, the temperature, and the treatment. So the standard method of doing this is to take your IP'd solution and leave it overnight with proteinase K treatment at something like 62 degrees, or without proteinase K treatment at 65 degrees. But you're waiting a long time for this to be done, and you're now taking the procedure to the next day instead of just carrying on during that day. We find that we did some experiments between either doing this overnight treatment or doing a quick treatment, and we don't find too much of a difference. And we actually usually, depending on the antibody, find the quick treatment is better. So here we're looking at a reverse cross-linking amplification, again, of that RPL primer set with the H3K4ME3 antibody. And what we see is that in purple here, we did a 15-minute at 95-degree treatment in a thermal cycler to reverse the crosslinks versus doing the exact same thing with a 2-hour treatment at 62 degrees with a proteinase K treatment to degrade the protein, or just the 2-hour 62 degrees proteinase K. And then both of these proteinase K treatments were followed up in the end with 10 more minutes at 95 degrees to deactivate the proteinase K before amplifying on the thermal cycler here, since the proteinase K can interfere with your reaction. So what we see is that the 15 minutes at 95 degrees was able to amplify more DNA than the other two treatments, and that still our negative loci down here did not amplify, and it did not change per treatment here. So we usually recommend just that quick 15-minute at 95 degree reverse cross-linking step. The next step, of course, is purifying the DNA so that you can use whatever assay you're going to use down the line. Now, most people, again, either do a phenol chloroform pellet step, which is fairly slow since you're having to mix the DNA in multiple stages with the phenol and the chloroform and letting it rest and shake it, and then having to pellet afterwards. It's fairly imprecise because when you are looking at your aqueous versus interphase versus organic phase down here, and you're trying to take off the entire top aqueous phase here, it's hard to get the entire phase out, either without leaving some or without accidentally picking up this interphase layer down here. We like to recommend silica columns instead. These are very, very quick to use. You can do easily 10 samples in 10 minutes. They're very efficient in recovering the DNA and also cleaning it from any other sort of contamination or small oligos, and the results are very reproducible. So we compared, again, just like with the beads, 6 different columns from major manufacturers here to see what the difference in efficiency of recovery of the DNA was after putting them through the columns per the manufacturer's guidelines. And what we saw here, again, a real-time PCR graph of amplification, and so we can see 6 different curve sets of doing this in triplicate, and it was just taking the exact same input DNA and then putting them through the 6 different columns in triplicate. And so we have 2 different columns here, 3, 4, 5, 6. And so when you quantify the differences between the recovery here, we can see columns 1 through 6 here and here, and the differences in the CT values. They're all compared to column number 1, which tested the best, and the worst was column number 6 here, which was over 5 cycles later than our first column. So what this equates to when we look at actual recovery percentage is that if we normalize our best column at 100% here, we can see that column 2 here pulled down about 93% recovery compared to 1. Column 3 did about 64%, and then we start to see a pretty bad drop-off. Column 4 only recovered 44%, column 5 only 13%, and then the very worst, only 3% recovery compared to column 1, which is about a 4,000% change here. So very, very important to make sure that you have a good column, recover the most DNA that you possibly can after purifying and then cleaning it up. The differences between 1 cycle really won't affect your experiments very much, but as soon as you start to get more than 1 cycle, 2, 3, 4, 5 cycles later, you're really pushing it as far as being able to quantify your DNA. So now looking at the very last step, which is actual amplification of your product that you pulled down. We have just a cartoon graph here of the very basics of what you would need to test it. First of all, we have our diluted input fraction, and that is in red here. And we always want to dilute the input to something like 2 or 1% from what it is at 100%, just because a lot of times your signal will be so strong at 100%, you will not get accurate amplification. I'll talk about that in a minute. But we've got our input fraction, we've got our actual antibody that we IP'd here in green, and then we finally got our no antibody control that we see has a large separation here. You always want to do your real-time experiments in triplicate, at the very least in duplicate, but in triplicate if you have the plate space to do it, if you're using a 96-well plate or a 384-well plate. There's also a sweet spot, I like to call it, for amplification in real time. A lot of people set their cycles for their thermal cycler up to 40 cycles or beyond, but usually once you start to get past the early 30s here of amplification, your data really starts to fall apart. If you are using triplicates, you'll notice that your triplicates are diverging from each other and not coming up at the same CT value. You'll notice that if you're running a standard curve, your standard curve starts to fall apart and your efficiency goes down. And so when you're looking at your data here, you really want to make sure that you're amplifying before that early 30s and not in your late 30s down here. And the same for amplifying too early. Again, I said with the input, you will lose that nice standard curve and the nice triplicates if it's amplifying too early, if it's amplifying at cycle 5 or so. The next thing is your primers. So you're using these primers to amplify your genes that you pulled down. And one of the things you always want to look at when checking to make sure that your run went okay is to look at a melt curve of your real-time experiment. And this is going to allow you to look for unspecific products. So this is just a sample melt curve here where we see as we increase the temperature, we split our products apart and they melted at the exact same temperature, which here is about 74.5 right here. Now, if you're having different products from contamination or your primers not being specific, not always, but usually you will see a different melt curve pattern to where there will be a different temperature that they're melting at or a different peak here that you're going to see. And a lot of times you'll see double peaks if you have more than one product that you're amplifying. The other thing to look at is to always run a standard curve. Like I said, run triplicates and run a standard curve in a serial diluted input to make sure that your efficiency is right around 100% for your reaction and that you have a good slope. If you are seeing things like 80% efficiency or 120% efficiency, you know something is going wrong. And you're going to need at least one of these input points here on the standard curve graph to use for that signal to input ratio that you're going to be using for your graph. So again, here we have our final graph after going through the chip procedure. We've used everything we needed to, the positive control antibody, the input fraction, positive and negative locus, to get good amplification and low background noise down here. So just to quickly talk about chip kits. So why use a kit? This is especially important if you are just starting in chip for the very first time or trying to restart it after a long time. In the kits, every component has already been validated and is hopefully the best in class here, so that you don't have to go through the same consumer reports type approach of testing 6 different purification columns and multiple companies' beads. You already know, hopefully, that you're getting a very good product that's been tested well and everything is there that you need. You can see that chip kits like ours here, the Chromatin chip kit, contain a lot of buffers, so having all these reagents and buffers ready to go for you is a huge time saver. So our chip kit in particular has the magnetic beads versus the agarose beads, comes with a positive control antibody that you saw today, comes with positive control primers, and purification columns here. This removes the entire troubleshooting phase. Since CHIP is such an involved and drawn-out procedure, you're not having to go back and second-guess which of your components failed or where you need to change something around. Hopefully, the first time you use a kit with having all the positive controls, you'll know that at least the experiment worked, and so the only thing that you'll need to tweak is your antibody of interest that you're using to pull down your specific target. The simple protocols are also very nice. A lot of the protocols, as I said out there, are very divergent from each other, and you're not sure which path to follow. With this kit, everything's already in place for you, and you know where to go and what to do. They're also very cost-effective with these kits. A lot of times, if you were to buy purification columns and beads and positive control antibody alone, you'd be spending more than the cost of the kit, and you're not getting everything else that's there. So they're very cost-effective. So that is it. Thank you very much, and I'll take any questions.
Okay, thank you, Eric. That was really interesting stuff and some really nice data there. So we do have some questions from the audience, so we'll just get in there. If people want to add some more in, then I'll try and take them as we go. The first one is from Matthew Fujita, and he's saying that are the beads 1 and 4, are those from beads 1 to 4, are those from different manufacturers? Is that correct?
Yes. So all of the beads that I have shown in the data here and all the purification columns were from different manufacturers.
Okay, and so why do you think those beads gave such different results?
That's a good question, and that's one thing I wonder myself. All the beads that were chosen, there's a lot of difference in beads when you're just looking at their properties. Some are what's called paramagnetic, where they don't retain any of their magnetic abilities afterwards, and some are just regular magnetic beads. They have different coatings on them, so these were all protein G or protein A and G coated. So it could be that the protein A or protein G that they're using to coat the beads just isn't as working as well or isn't coated as well. I looked into uniformity of the beads when testing the beads. Some manufacturers like to promote how uniform and spherical their beads are, as you saw in that picture that I put up there with the beads, how perfectly circular and equal size they all were. I didn't seem to see much of a difference between that. I looked at multiple beads. I tested probably 2 to 3 times as many beads as I had data for here, and looking at the different beads under the microscope, some were perfectly spherical and worked great. Others were also looked just as perfectly spherical and uniform and performed terribly. They didn't pull anything down. So I don't think it's shape. I think it probably has to do with the coating, the protein A and protein G.
Okay, that's quite interesting, isn't it? So a couple of the attendees were Lee Barrett and Tomohiro were asking if you could tell us who the manufacturers of the beads and columns are, but I guess that is your kind of you wouldn't want to share that, I guess.
Yeah, I thought that question would probably come up, and I don't want to badmouth any other companies out there, so I don't really want to do that. The only thing I can say, I guess, is that all the top performers in the fields that I tested are components of what we use in our chip, in the Chromatin kit, and we also sell separately from that, but I don't really want to disclose who the bad performers were.
Sure. Okay, on to another topic. So from Zoe, for the immunoprecipitation, is it better to use the antibody against your protein of interest, or can you also use antibodies against fuse tags such as his tags?
That's a good question. You can use the tags. You should have a lot more pull down from that versus doing a modified antibody or transcription factor, but it should work just as fine, and it should work actually even better. You just have to be really careful about your background again and making sure that your no antibody control or IgG only control isn't pulling up anything with it.
Okay. Okay, so also on the antibodies, Benjamin Taylor is asking us quite a long question, so bear with me, but it's quite a good one.
Sure.
Benjamin says, we see no enrichment in our chip assays, and the antibody we are using doesn't perform well when tested in a Western blot. My supervisor argues that what's happening in the Western blot doesn't necessarily reflect what's happening in the chip assay. And he's asking, is this a valid argument? Is an antibody that doesn't work in a Western likely to work in a chip assay?
I think you're both right in that in a Western blot, most Western blots we're looking at a denatured and reduced protein. So in a chip assay, we're looking at something that's in its native conformation. It's not denatured or not reduced. So if it is an antibody that isn't performing well in Western blot alone, you might want to look at it in a non-denatured and non-reduced native state and do a native page, or possibly doing a combo, doing the pull down. Like I said, if it works in IP well, then usually it works in chip well. So if you use that antibody that isn't working well right now in a native IP, and then maybe even used a second antibody that is known to work well in Western blot for the same target, and you see a pure, clean result, that you'll know it is working well and possibly will work well for the chip in the native state.
Okay. Getting some questions just coming in, so I'll try and keep up. Okay, so we have one here from Amy. This is on the automation. So she's asking, how long does a standard chip protocol take, and does the precipitator machine make it faster?
So that's a good question too. So with like the kit that we developed here for the Chromatin chip, we did all these experiments to try to have the best components to cut down on time as much as possible. Like I said, with that reverse cross-linking phase and having to do an overnight part of the assay and using up a lot more of the time, these assays can be very, very time-consuming and take multiple days to do, depending on your protocol. So the only thing that I always recommend to do overnight is the antibody incubation with your chromatin overnight, but everything else can be done very quickly. Usually about less than a day and a half you should have results. So the precipitator helps a lot in cutting down on the time of having that automated phase, especially if you're multiplexing and doing more than one sample at once, because with the precipitator, like I said, you can use 16 different samples, so you are moving the beads from one well to the next within 15 seconds for 16 samples, whereas otherwise, even using the magnetic bead technology, which will make things go a lot faster regardless, you're still having to transfer those beads into different wash buffers, one at a time for each of those 16 different samples.
Yep, I guess it'll make the whole thing more reproducible as well, using the automated protocol.
Yeah, I always include a positive control antibody with all of our tests here for doing chipping to validate any of our antibodies, and it's really amazing how consistent the results are. That amplification that I'll see for that same primer set and that same antibody will be within 2% every time over and over again, and that's also very important if you are trying to compare different treatments that you didn't do at the same time to show that the results are similar.
Okay. All right, so here I also have a question here from Denise Kimbra, and she's got a specific technical problem that maybe you can provide some input on. So she's saying that when she's consistently getting false positives from negative controls using agarose beads. Could it be the washing that is the problem, or the type of beads being used? So she's saying how beneficial is salmon sperm pre-clearing with magnetic beads, but I guess you've said that you don't need a blocking step with the magnetic beads, so maybe you would recommend that she switches from agarose to magnetic.
Right. As I put in that one slide where the two amplifications came up right next to each other, that's the high background that you can see either with the magnetic or with the agarose beads, but it is more typical of the agarose. And so if you want to stick with the agarose and you're not doing a blocking step, I definitely recommend you do a pre-blocking step of the beads, and you really should be having that thousands of % less amplification of your no antibody controls versus your actual antibody IP, because I have looked at similar data from people that were using beads that they were saying they had a significant increase in a gene versus a no antibody control, but the fold change was 1.5, and the fold change should be hundreds and hundreds and hundreds apart.
Yeah. Okay, so back to a more general question from Ziba Wunderlich. Sorry if that's not pronounced correctly. But she's saying, are there good guidelines for how much input material, such as tissue or DNA, you need for each step of the protocol? Do you provide those in your protocol?
We have, for our chromatochip kit in our protocol, we have a basic guideline walkthrough example where we're just using HeLa cells and give a 10-minute fixation time for 10 million HeLa cells. But this can vary greatly depending on your cell type, and especially if you are using tissue, and dependent on how you are fixing that tissue, if you're doing a fresh perfusion of the animal versus post-fixing after dissection. And more than anything, it's one of those things where it truly is the experimental phase where you're just going to have to see what's best for you by testing different fixation times and different cell quantities. But you will see the difference at the shearing step right away in the very beginning of how well it's shearing, and that's really what you're optimizing it for more than anything, to get that perfect smear between 100 and 1,000 bases. So if you don't have a good smear, you don't have good fragmenting, stop right there. Don't continue on to the rest of the protocol. But we have a lot of examples, so calling our tech support can also help you as a shortcut to getting where you need to go there too.
Great. Okay, and also on that topic, a couple of people are saying that they don't have access to sonicators, and so they're asking, is there any problem with doing an enzymatic digestion at that step?
No, it's just a different approach. It's not a problem. It's just more, what do you want? So like I said, with the enzymatic step, you're going to have evenly cut pieces, and so if you want a more truly random shearing, depending on where you're placing your primer sequences around, your primer sequences may be right where that restriction enzyme is cutting. It may be cutting it too short where you can't get the full amplicon, whatever amplicon size you're looking for. I usually recommend for RT-PCR right around 100 base pairs. Whereas with the sonication, you know that it's random, so you should be getting fragments right in that region of the amplicon that you're looking at, no matter what.
Okay. So we can just squeeze in one last question, and we'll go for the one from Tomohiro Masuda, and he's asking, is there any way to find out which antibodies will work for CHIP? Is there a list of validated antibodies somewhere? that can be accessed?
Yes. So most companies will grade their antibody and call it chip grade or chip validated. This is also, you don't want to use it as a misnomer. You don't want to say that if it's not chip grade or chip validated that it's not good for chip. It just means that the company hasn't had the time to validate that particular antibody for it. So if you see that the antibody is well validated in other terms, if it's working in IHC or ICC, so that you know that it's working in its native conformation, if you see that it has dot blots or any sort of peptide arrays associated with it from publications, especially for these modified antibodies, so that you know that it's absolutely specific, because that is a huge problem where a lot of these modified antibodies, especially the histone ones, will be picking up more than they say they are. And like I said, if it works for IP for you, if you need to test it and it works in IP, then there's a really good chance that it's going to work well for you in chip.
Great. Okay, thank you very much for that, Eric. We'll wrap up there. Apologies to everyone who we didn't manage to get to your question. But I thought that was a really excellent presentation and nice question and answer session too. So thanks, Eric. And thanks also to the audience for taking the time to attend and listen in. If you've enjoyed the seminar and you'd like to view the video recording of the session, please visit the seminars page on bitesizebio.com, and it should be available there within the next 24 hours. And there you can also see the other webinars that we have lined up for you over the coming months. There's a few good ones in there, so please do take a look. And so we'll leave it there, and until next time, good luck in your research, and goodbye from all of us at Bitesize Bio.